We're getting closer and closer to some exciting bugs. I actually totally forgot to upload the next slides. I will do that. I will give you a little break so you can go and print it. Don't worry about that. It's going to be really quick. Now, we're going to talk about 2 gram-negatives now. The Yersinia and Francisella, and 2 species can be a considerable threat to the human health. Yersinia pestis and Yersinia enterocolytica. Yersinia enterocolytica. I don't want you to focus on anything particularly specific regarding the virulence factors, but Yersinia enterocolytica causes enterocolitis, okay, gastroenteritis, and it is the zoonotic infection, okay, that can end up on the human food and can be acquired from the food by humans. Less common transmission is from domestic animals that are not entirely domesticated. What I mean by that is, if your dog or your cat lives sort of a feral life, runs outside a lot, spends time in the forest, and so on and so forth, then it has chances to acquire Yersinia enterocolytica from the wild animals and bring it to the house. And then it can be further transmitted by contamination of the food to humans. Does that make sense? Yersinia enterocolytica is what is sometimes called orphan disease because it is often overlooked as the causative agent of gastroenteritis. There was a case when a woman adopted a child, really small child, something like an infant pretty much, something about a year old, and child was placed in the foster care because of the neglect from his biological parents. The neglect was that child pretty much slept on the floor next to dogs and cats in the household. And I have nothing against dogs and cats, but that's not the way you treat a child, the infant. So he was placed in the foster care, and the child he wasn't really healthy, he had diarrhea. Pretty much chronic diarrhea. Bouts of diarrhea going back and forth, and this woman visited a number of physicians trying to figure out what is wrong with the kid. None of the physicians could help her. They checked the child for a number of different diseases, but nothing came up positive. And she started to read about different orphan, overlooked infections, and she found Yersinia enterocolytica. She came to a physician and said, "Could you test him, please, for that?" He said, "Sure." Tested, and he came up positive. So he obviously acquired infection from the dog or a cat, because in that household the animals were not kept at the house all the time, and then developed that kind of an awkward manifestation. It was an acute disease with bouts of acute disease. So his immune system was not strong enough to completely eliminate disease, but it tried to keep it under control, so that created this bouts of disease. That makes sense? Now, Yersinia pestis. Another name is plague. One virulence factor that I want you to focus on, I want you to know. Yersinia pestis loves iron. I believe we have discussed the case of the professor from Arizona State University who acquired vaccine-derived strain of plague. Have we? You don't recall? Lovely case. Just brilliant case. Gentleman in his, I think he was like 50 something, comes to the emergency. It's an actual case described in the morbidity and mortality weekly reports by published by CDC. Guy comes up to the emergency room saying that he feels bad, like doesn't feel well, malaise, very high fever, something like 104, asks to help him, asks the doctors to do something. Doctors evaluate him, nothing of a particular interest. They say it's probably some sort of virus. Go home. Take some ibuprofen. Arizona State. The guy comes home. Next day he returns to ER, this time using the ambulance vehicle because he cannot drive, because he's confused, has spiking fever, 106 or something. He can barely speak, he's delusional and whatnot. So they admit him to the hospital and start broad spectrum antibiotics. I don't remember which ones. I think it was tetracycline and vancomycin. Doesn't really help. In 24 hours he dies. 24 hours after his death, results of the labs come in. Turns out the guy has a plague, like real plague. Well, when everybody stopped freaking out, they started to investigate what's going on. Turns out the guy that died was a professor of microbiology at Arizona State University studying, guess what? Plague. He was studying a vaccine strain, attenuated strain that is not supposed to kill humans. And when they compared the strain that he had and the strain that he studied, they matched. So he obviously inoculated himself while working in the lab. So that's number one. Sort of a little digression. Bless you. Whether you work for 1 month, or you work with 101 months, or 101 years in the lab, safety is the safety. Probably it was, oh, I've been doing that my entire life. I can ditch the gloves. No, you cannot. Safety rules are safety rules. So he probably violated them. But one question still remained open. How he got so sick because, well, while being infected with a supposedly attenuated strain, weak strain that should not cause disease. So they started to dig his medical records, and it turned out that the guy had the condition known as hemochromatosis. Does anyone know what it is? It's abnormally high levels of iron in the blood. So a person constantly has abnormally high levels of iron in the blood. Plague loves iron, just loves it. So elevated iron levels promoted growth of otherwise benign strain, leading to vast immune response, just out proportionally stimulated immune response, and the guy died. And technically any person with hemochromatosis is more at risk for the septicemia, for the replication of bacteria in the blood. I specifically stressed out Arizona State University. Reason for that is that Arizona, New Mexico, California, Nevada, Utah are endemic for plague. Well, should we say endemic or sporadic? It's hard to say. Let me put it this way. Plague is zoonotic disease. Okay, which means that it persists in the reservoir of rodents. And transmission between different rodents, whether those are rats or mice or groundhogs or prairie dogs, is via fleas. However, if your domestic animal, like if your dog runs into the prairie and then accidentally and chases the prairie dog and picks up a flea, so flea can jump, right? Picks up a flea and comes into the house and fleas can jump on you, chances are not very high, but there are some. So every year 1-2 cases of plague are registered in the United States in that southwest region. Okay. So fleas transmit plague to humans not frequently but regularly. Whether it's a sporadic infection or endemic infection, it is not of a major concern, really, now. It happens every year. And there are more than one enzootic region of plague in the in the world. The United States is southwest. In Russia, it's Volga River. In Kazakhstan, it's Volga River. So there are spots of plague in different parts of the world. You have to appreciate, though, that this disease can be fairly easily treated, okay, with antibiotics. A tetracycline is hugely effective against plague. Now, what are the forms of the plague? Typical form, what we call it bubonic plague. You've heard that name before, I bet. So it is called bubonic because up to the initial infection, following the flea bite, bacteria migrates into the adjacent lymph nodes, causing the formation of so-called bubo. Lymph node, replication of bacteria in the lymph node, leads to necrosis of the lymph node and swelling. It's a black necrotic lymph node. Bubonic plague has pretty high mortality if untreated, up to 70%. Okay. People with bubonic plague can transmit the disease by a respiratory route. And if plague is acquired by a respiratory route, the mortality can reach up to 90%. Septicemic plague, which is replication of the plague in the blood, is practically 100% lethal. Okay, so whether it's a development of, if plague leads to septicemia, it's a death sentence. Right? How plague came to Europe is still sort of, well, we know who brought the plague to Europe. In the famous outbreak of 14th century, 1300s, it was Arabs. They were, well, two implications. One, Venetian tradesmen. They were sailing to Asia for, exchange of goods and stuff. And they apparently brought some rats with them. Okay. And second, Arabs tried to capture Venice, and they were first to use biological warfare. They had their own people that died from plague, so they used them as the, what would it be, vehicle, fomite? I don't know, fomite probably. They just catapulted them into the city, behind city walls. So they pretty much spread the disease. Arabs already knew that it's highly contagious. And I mentioned that initial epidemic of plague killed, by some estimates, to half of European population. And second was not that bad, and the third one was even less contagious. Probably because we acquired some genetic resistance. All the weak ones were killed. If you would Google plague or plague doctor, you'll find this weirdly looking picture of the guy with the bird mask. Have you seen those? The belief was that you can scare the plague by wearing this mask, which was kind of a wild belief. But interestingly enough, those doctors, on average, survived longer. So it took them more time to acquire plague. The reason? Yeah, they wore masks. It was essentially, it protected them somewhat against the respiratory transmission. It wasn't, of course, perfect, but it was pretty much first personal protective equipment, first PPE in the world. Another disease that I want to mention is tularemia, caused by Francisella tularensis. For Francisella, one of the major virulence factors that I want you to know is that it can successfully survive the macrophages. Can be acquired by a variety of ways, starting from the contact to vector to respiratory route. The contact tularemia can occur if you skin the diseased animal, like skinning a diseased rabbit. Rabbits are the probably major reservoir of tularemia in the wild. It starts with the lesion on the exposed, the affected area, the skin, and then progresses to the systemic disease. The form of tularemia that is acquired by cutaneous route, the contact route, isn't, well, it's dangerous, but it isn't that as lethal as the pneumonic form of tularemia. Now, a pneumonic form of tularemia is acquired when bacteria become aerosolized. You may say, wait, how can you possibly aerosolize the rabbit? There were cases recorded when people were riding the lawnmower. There was a dead rabbit in the grass, and they didn't notice, and they pretty much aerosolized it, inhaled it accidentally, acquired tularemia. Okay, mortality if untreated can reach up to 50%. It's a BSL-3 pathogen. There is a vaccine for both plague and tularemia, but we don't use them for general population because we are not exposed. But for instance, a former colleague of mine who works on tularemia, they were going to work with the wild type strain, the pathogenic strain in BSL-3 facility, and they all had to get vaccinated with live attenuated vaccine. Okay. Actually, that's the first microbe, tularemia, that was considered for biological warfare usage. The first one that I mentioned was Bacillus anthracis. That's the second one, and allegedly Soviets used tularemia, tried tularemia as biological warfare in the trenches of Stalingrad against Germans, because German documents reported the outbreak of tularemia in Stalingrad. There were like 200 people that died from this disease. I don't really know if that, not that I'm trying to protect, but it wasn't the clear experiment. Why? The conditions in that epic battle were so poor. People had to be in the trenches, and it's prairie, so there are plenty of rodents and whatnot, you know, animals that can potentially carry tularemia. Whether it was an actual biowarfare or it was just an outbreak caused by the local, you know, population of animals, who would know? Pseudomonas, Legionella, and HAEMOPHILUS influenza. Pseudomonas aeruginosa. This is the microbe which name should pop up in your mind when we talk about hospital-acquired infections. Pseudomonas, it can be acquired from environment. It's pretty common in various community settings. We will discuss which community settings. But it's notorious. It's known to be absolutely notorious for its ability to cause hospital-acquired disease. And it has a bunch of virulence factors as well. I want you to focus on one big virulence factor that is called drug resistance. Pseudomonas is the great example of the bacteria shows a number of mechanisms to withstand the action of antibiotics. Among them, it contains genes for beta-lactamases, rendering this microbe resistant to, say, cephalosporins. It contains the genes, altered gene for DNA gyrase, which means fluoroquinolones are not going to act on it. It can destroy aminoglycosides and chloramphenicol, protecting its translation. People try to count all the mechanisms. I think different strains, if you will put them together in one hypothetical horrible strain, it would be resistance to everything that we have. Okay. In terms of the disease, though, Pseudomonas can cause a variety of infections, starting from the skin infections to the urinary tract to the lower respiratory tract infections. In terms of the skin infections, you may have heard the names like green nail syndrome or hot tub rash. They both kind of self-explanatory. Okay. Hot tub rash can progress, actually. Skin infection with Pseudomonas can progress to the condition known as, I don't remember if I mentioned it here. Yeah, no. Erythema gangrenosum. Don't write it down. So it's a small necrotic lesions in the subcutaneous tissues. And the problem with those lesions, if they're left untreated, they can further progress to necrotizing fasciitis, and that is practically either losing the part of the body or death sentence. However, in addition to urinary tract infections, Pseudomonas is the very important cause of lower respiratory tract disease in patients with cystic fibrosis. It's a major infectious cause of death in patients with cystic fibrosis. Pseudomonas becomes practically resident in the mucus of lower respiratory tract in cystic fibrosis patients. Remember, in cystic fibrosis, there's a mutation in the gene that encodes one of the chloride channels. So patients produce an enormous amount of mucus that they cannot clear, and that mucus provides great breeding grounds for various bacteria, but Pseudomonas is number one. And patients with CF have to be on antibiotics always, and it drives the resistance, antibiotic resistance in bacteria farther. However, recently, the clinical criteria for placing patients with CF on antibiotics were reconsidered. Previously, if the patient was found to be positive for Pseudomonas, it was a warrant to start antibiotic treatment. Now, if cystic fibrosis patient is positive for Pseudomonas but has no clinical symptoms, then treatment is sort of delayed or postponed. He's just being observed. That's the attempt to reduce the use of antibiotics, obviously to reduce the development of resistance in the bacteria, right? This is the short list where you can find Pseudomonas. It's pretty resistant, so you can find it even in the solutions of disinfectants. Walls. You can find it on walls. There was a proposal. One lab developed the paint, antibacterial paint, and they suggested to use antibacterial paint to reduce Pseudomonas load in the hospitals. And nobody decided to go forward with that because of the resistance. Nobody wanted a resistance to get developed. Does that make sense? So that's drug resistance and major conditions associated. So cystic fibrosis patients, the pneumonia, green nail syndrome, hot tub rash, skin infections. Legionella pneumophila is the gram-negative bacillus that is a great example of non communicable disease. It was first isolated from patients that died from pneumonia after acquiring this bacteria during the American Legion conference somewhere. I don't remember where. I think it was 1976. Just several dozens of people went down with pneumonia, and almost all of them died because they couldn't figure out what's wrong with them. And it turned out that was gram-negative microbe, Legionella pneumophila, that they acquired from improperly maintained air conditioning systems. Legionella in nature is the pathogen of amoeba. It infects amoeba, replicates in amoeba. And if you have air conditioning system with water, condensed water that drains, if there is a little bit of a delay in draining, if there's some water, then amoeba start to reproduce, and where is amoeba, there is Legionella. So people acquired Legionella. It pretty much was accidental infection. And Legionella ended up in the lungs. There are cells in the lungs that look a lot like amoeba. They feed like amoeba. They move like amoeba. The phagocytose like amoeba. The phagocytose in the lungs, in the alveoli. Which cells phagocytose? Macrophages, alveolar macrophages. Legionella infects alveolar macrophages. The only problem, it cannot go anywhere else afterwards. It's stuck in the lungs. So people with Legionnaire's disease cannot transmit the infection to other people. Okay? So you can acquire it, but cannot transmit. There were some outbreaks of Legionnaire's disease, pretty much that happens every year. In Cedar Point, I think last year was one. There was an outbreak in New York last year too. You know this water sprays that kind of make like a cloud of water that to cool you down during the hot summer day? So these things, they usually have a lot of Legionella, and they're a major source of outbreaks because people can inhale that water. It's aerosolized enough so you can inhale it, gets into your lungs and leads to pneumonia. It's fairly easily treated now, so it's not mortality is high if untreated, but we now are aware, okay, and we can treat it pretty well. Haemophilus influenza. It's a fastidious bacilli for which the main virulence factor is the capsule. Okay, so know about that. It is often present in the upper respiratory tract microbiome, in the pharynx, the ear, the pharyngotympanic tube, and it's a very common causative agent of the middle ear infection, the otitis media. However, if it migrates into the bloodstream and ends up in the meningeal circulation, you've got meningitis. To prevent this, we have very effective HIB vaccine. One thing that I want to remind you, Haemophilus influenza has nothing to do with influenza. Okay? It's not a flu, it's a completely different infectious agent. Okay? Campylobacter jejuni and Helicobacter pylori. The name for the species, jejuni and pylori, shows you where you're going to find those microbes. Helicobacter jejuni is the most common cause of bacterial enteritis in the United States. The number of cases is estimated to be about 300,000. Okay. I'm not gonna focus on any virulence factors. Campylobacter is the common disease acquired from poultry, turkey, raw chicken, water, contaminated milk. It can be destroyed by pasteurization, but we do not pasteurize the chicken. It has been implicated in the Guillain-Barré syndrome, which is an autoimmune condition when antibodies that your immune system produces against the pathogen start to recognize your own tissues. Guillain-Barré syndrome manifests as the flaccid paralysis. In majority of cases, patients with Guillain-Barré recover completely, but in rare occasions, the paralysis or limb weakness remains, and that is a debilitating condition, chronic debilitating condition. Disease is largely self-limiting, which means no additional treatment except supportive treatment is required. So this microbe, Campylobacter jejuni, can be found in the small intestine, in the jejunum. Helicobacter pylori can be found in the pylorus, part of the stomach, the distal part of the stomach. And Marshall and Warren, two Australian scientists, noticed that people with gastric ulcer, all of them, have Helicobacter pylori. So they saw a very strong association. And they suggested that gastric ulcer is caused, maybe indirectly, but caused by Helicobacter pylori. Well, how can you prove it? Think Koch's postulates. What do you have to do first? Yeah, but first, even before. The microbe. Well, let's start at this. Turns out this is the very picky microbe. It's microaerophilic, capnophilic. Remember Goldilocks? So they had to figure out the optimal concentration of oxygen, optimal concentration of CO2. It took them months to finally grow Helicobacter pylori. And they grow it, but it turns out it doesn't cause anything in mice. They became sort of desperate until I never remember who it was. Either I think it was Warren who took a beaker of liquid culture of H. pylori and drank it. We didn't have this much amounts of LB broth. It stinks. Let me tell you something. The liquid broth for growing microbes, it doesn't really taste good. It tastes lousy by itself. Well, throw some microbes on top of that. Doesn't make any better. But he drank the entire beaker. Guess what? In a few days, developed all classical symptoms of gastric ulcer. Endoscopy showed that he had gastric ulcer. And they did a biopsy of his stomach. And the biopsy was textbook gastric ulcer. So he pretty much did the Koch's postulates on himself. They isolated it from him. Ended up getting Nobel Prize in 2005, if I'm not mistaken. And now it is established that H. pylori can cause gastric ulcer. So what happens? The microbe decreases the production of mucus. Okay, it breaks down the mucin, major protein component of mucus. Okay, And to survive the acidic environment, it also produces urease that neutralizes the acid, right? Now, its adhesion to endothelium leads to the local inflammation. Local inflammation means that white blood cells will migrate from bloodstream to the site of inflammation and will try to repair it. However, microbe doesn't go anywhere, so it's constant local inflammation, which leads to the tissue destruction. You can see on the right hand side of that right picture, it's the ulcer. The left picture is the healthy pylorus of the stomach. Okay. There were some weird associations. There is an association between the gastric ulcer and gastric cancer. So the idea is that gastric ulcer is the first stage, if I may say so, towards the gastric cancer development. Kind of falls into the current hypothesis that cancer is the inflammatory disease. Chronic inflammation, chronic repair leads to eventually to the formation of malignant tumor. Interestingly enough, Helicobacter pylori was shown to have inverse association with the adenocarcinoma of esophagus, which sounds counterintuitive. But one of the possible things to think about, Helicobacter pylori should reduce the acidity of the intestine, of the stomach. Reduced acidity will lead to the less gastroesophageal reflux, and gastroesophageal reflux itself is associated with esophageal cancer. Another interesting inverse association: kids with H. pylori less likely to develop asthma. They're more likely to develop ulcer, but less likely to develop asthma. Your pick. And the how do we actually know that it's infectious disease? When we treat patients with ulcer with antibiotic and proton pump inhibitor to reduce the acidity, okay, then they recover much faster. Antibiotics help compared to non-antibiotic treatment, okay. Bismuth as a treatment, the component of Pepto-Bismol. You would read the name on the label. It says sodium bismuthate. No. It's a salt of bismuth subsalicylate. Yes, it's a bismuth subsalicylate. So bismuth itself reduces the inflammatory effects of Helicobacter pylori. Subsalicylate has two effects. Subsalicylate is the relative to the salicylic acid. You know the common name for it? Aspirin. So it's sort of kind of aspirin, reduces inflammation. And second, it has non-specific antibacterial activity, like general antibacterial activity. So when you have a cocktail of those drugs, you treat both the replication of the microbe and you reduce inflammation. That makes sense? So again, what you have to know, there are too many virulence factors. Don't focus on virulence factors. The gastric ulcer and the idea that treatment with antibiotics is more effective kind of shows that H. pylori is the infectious disease. And yes, it can be transmitted from human to human. Okay. And yes, there are people who have H. pylori but do not have ulcer. So colonization is possible. Okay. Treponema, we're going to talk about this. I'm going to give you a break, upload some more material. Sorry that I didn't do it early. It completely escaped my mind. It's going to be not very many slides. We're going to start talking about viruses today. Treponema, Borrelia, and Leptospira. Those are spirochetes. I put them in the gram-negative group. They are technically gram-negative. However, detecting them using gram stain is extremely difficult. The reason for that is the shape. They all are spirochetes. So you see the dark field microscopy on the left, phase contrast on the right. Think about this. They are so thin that seeing them using regular gram stain and bright field microscopy is extremely hard. That makes sense. Treponema. Genus Treponema can be found normally in the human mouth. Various types of Treponema species are a part of your normal oral microbiome. The featured organism, Treponema pallidum, though, is the infectious agent that causes syphilis, sexually transmitted disease. I'm not asking about the virulence factor, but we're going to discuss different stages of this disease. First of all, syphilis is transmitted sexually. Can syphilis be transmitted by contact? Like if a person with syphilis gives a handshake to a person that is healthy. No. The only way syphilis can be transmitted not via sexual contact is when people share things like underwear or towel. There are recorded cases. It's not frequent, there are recorded cases. Other than that, it's not transmissible by contact. So, after the initial infection, syphilis causes the lesion on the genitals that is called a chancre. Guess who notices syphilis more frequently? Men or women? It's men, because it's more obvious, okay? Mucous membranes are more exposed and men observe them more frequently than women. So in women, often the first stage of syphilis goes unnoticed. And other than the chancre on the mucous membranes, there are no other manifestations. Then, and first stage is infectious. After a while, the chancre goes away, and after some time again, the secondary syphilis ensues, which is manifested with the rash. You cannot really see it here. There are pictures. If you would just Google, due to the copyright restrictions, I try to avoid copyright conflicts as much as possible. I mean, if you Google, there are no copyright conflicts. Rash can cover the entire human body. It's very noticeable. It's bright, dark red spots. And flu-like symptoms. A friend of mine who graduated from medical school, he had to write a term project on secondary syphilis. And he had a little book. It's called like a secondary syphilis for doctors. Almost sounded like user's manual. And I just glanced through the pictures. Believe me, it's not only rash. It gets much worse. And my only question to patients who were photographed, a hypothetical question, would be, do you have a mirror at home? Did you see that something is wrong? Grossly wrong. So there are lesions, huge lesions on the genitals. So they're like half eaten away. I mean, notice it. It's not right. Go to the doctor. Check it out. Secondary stage is also infectious. Then after a while it goes away, and then maybe sometimes after years and years, the third stage starts, this tertiary syphilis. Tertiary syphilis is non-infectious, but in terms of the symptoms, it's probably the worst, because at this point syphilis migrates into the virtually all tissues, starting from the brain to the soft connective tissue, to the hard connective tissue, to the heart, and initiates the destruction of those organs. Neurosyphilis, for instance, often manifests, especially if the spinal cord is affected. It is manifested as the lack of the knee-jerk reflex. Cardiovascular syphilis leads to the cardiovascular disease and, cardiac insufficiency. Infection of soft tissues is often manifests as so-called gummas. On the pictures that are rather disturbing, gummas sometimes can be seen as giant enlargements in the head, on the face, disfiguring enlargements. Another thing that can happen with the syphilis infection is not outgrowth and inflammatory outgrowth of the tissues, but rather destruction of the tissues. You can see here a gumma that is on the person's nose. In severe cases of tertiary syphilis, the external nose, the bridge of the nose, nary, and the nasal septum, they are completely destroyed. So the person doesn't have an external nose. That makes sense? They may eat out the soft palate and so on and so forth. Now, to give you a little chilling perspective, first of all, we still don't know where syphilis came from. There's an idea that it was sort of a gift back from Native Americans. We gave them a wonderful thing. It's called measles and smallpox. And supposedly they gave us syphilis back. So it's quite a fair exchange. It's not entirely clear, because there are diseases called Basel and Yaws that can be found in Africa and Arabian Peninsula. They are sort of milder forms of syphilis associated largely to the disfiguring malformations of the face. So maybe it was in the Old World, even before Columbus sailed to the Americas. Now, to give you a perspective, if somebody acquired syphilis in 19th century, that was it. There was no treatment. So it was supposed to progress all the way to tertiary syphilis, with all the malformations, tissue destruction and whatnot. If person wouldn't die of anything else before that. So this is a pretty grim perspective. Okay, now, for me it was always fascinating how people were scared of diseases that transmitted by sexual route. In my personal speculative opinion, there are two things. One, the idea that you can acquire something deadly by the process that is supposed to bring pleasure. Okay, we had a saying that all diseases are because of the nerves, and only syphilis is because of the pleasure. And second, sexual encounter was sort of a shameful thing for a long time. It was the thing that people just didn't talk about. From the microbiology perspective, if somebody can explain me what's the conceptual difference between sexual transmission and respiratory transmission, I would be eternally grateful. I don't think there's any. It doesn't matter. It's all disease. So now we can treat syphilis very easily. The first drug against syphilis was salvarsan by Paul Ehrlich. We discussed that it was effective, but side effects were pretty horrible. And then Alexander Fleming, penicillin. I don't know what people were more happy about, that you can treat wounded soldiers, or you don't have to be afraid of the syphilis anymore, because penicillin was fantastic. Of course, since then, syphilis acquired resistance to penicillin largely. We still, though, can treat it with the various penicillin derivatives, like ampicillin, amoxicillin. Okay, so it's still a reportable disease. So if somebody is diagnosed with syphilis, the disease is getting reported to CDC. By the way, back home, if somebody got diagnosed with syphilis, forget about HIPAA, forget about patient rights and all of that. Relatives, colleagues, the entire social network will get notified because of the potential transmission. Borrelia burgdorferi, causative agent of Lyme disease. It's transmitted by the Ixodes ticks, which in nature normally cycle through the rodents and deer. And this is the great example of humans being accidental hosts, because this bacteria normally cycles through rodents and deer. When it gets into the humans, it's a dead end. It's also called a dead end host, because from humans it doesn't go anywhere. So after a tick bite, several days after a tick bite, the characteristic bullseye rash, okay, or erythema, what's the name? Erythema migrans, sorry, erythema migrans, bullseye rash, develops at the site of the bite. And it's very, very typical symptom, sign of the Borrelia burgdorferi infection. And if this sign appears, you should go to the doctor, you should explain that it was a tick bite, and the doctor will give antibiotics, and you should start treating the disease immediately. Because if you don't, eventually microbe migrates into the joints, causing arthritis. Also, it causes cardiac problems, because it can infect cardiac muscle. So therefore problems with the conduction system of the heart and different types of myopathies. And it can actually migrate all the way up to the brain, causing meningitis, encephalitis, paralysis in severe cases. The arthritis is the most common complication of Borrelia burgdorferi infection. And the problem, some people just wait thinking, it's going to be fine. The problem is when arthritis starts. Treatment at this point may take up to 4 months, because it hides in the joints really, really well. It's really hard to get rid of it. And finally, leptospirosis, Leptospira interrogans. It's a zoonotic infection that can be found everywhere, okay, in the United States, in Korea, everywhere. It resides in the animals, domestic animals. And when transmitted to humans, usually manifests as the mild fever with myalgia. But if it becomes complicated, if it becomes systemic, then it can cause so-called Wales disease, characterized by the failure, kidney failure, liver failure, and massive vascular inflammation. Okay, for those 3 microbes, to kind of sum up, I am not requiring any virulence factors. You gotta know the transmission route for every, as I mentioned. Leptospirosis is zoonotic, borreliosis is zoonotic, and Treponema pallidum is the human-to-human, the sexual transmission. Diseases that they cause. For syphilis, know the three stages. Okay, 1, 2 infectious, 3rd non-infectious stage. Okay, is that clear? Okay, let's take about 10 minutes.